bats are one of the most functionally rare mammals, being the only ones to achieve true flight. In metropolitan France, there are 36 species of bats, all of which are insectivorous. They use echolocation by emitting ultrasonic signals and then analyzing their return echo to identify potential prey and landscape structures. They typically forage in insect-rich habitats such as forests, hedges, and rivers. These critters play an essential ecological role and are bioindicators of the health of an ecosystem. They also provide key services that prevent economical losses, such as fertilizing the soils with their droppings, and biological control of agricultural pests and of vector arthropods. As a result, all French bats are nationally protected, and 19 species are considered of conservation priority. Unfortunately, they greatly fall victim to civilization, by way of light pollution. Artificial light at night, ALAN represents traps or barriers along the path bats take during their nightly activities. It also masks natural signals followed by bats that accommodate rhythms in the natural environment. Bats typically respond to ALAN in two ways. They avoid it or they tolerate it. However, all are affected by light pollution, when considering the context and the scale at which they are studied. Fortunately, light reduction strategies exist. In Ariège, light pollution trends have decreased in recent years, and part night lighting (PNL) has been tested in a few communes. This measure consists in switching off outdoor public lighting for a portion of the night. But did bat populations in Ariège benefit from these light reduction efforts? To check this, we will look at bat activity in a two-part complementary study. First, a long-term large-scale setting, looking at the whole Ariège department across 6 years. And then, to explore causality, a short-term small-scale setting, targeted on 6 Ariège communes subjected to a PNL experiment. As they influence bats' responses, we are also interested in looking at landscape configurations and bats' echolocation ranges. For this, we will use passive acoustic monitoring, a non-invasive method that collects recordings of bats' echolocation calls overnight. These recordings will be used to identify bat species, then to evaluate their activity and behavior. Associated with PAM, we will perform GIS mapping in order to connect the recordings to specific geographical locations, landscape features, and light parameters. At smaller scales, we expect PNL to benefit light-averse bats by reducing predation risk, though opportunistic species may decline as insect densities drop around street lamps. Across larger scales, meanwhile, we expect overall bat activity to rise as ALAN levels decrease, particularly in less urbanized habitats. We hope our findings pinpoint optimal landscape configurations for mitigation, encouraging policymakers to target conservation efforts efficiently for both humans and wildlife.